Hello friends, welcome to my channel. This is Medico Link and I am Dr. Prashant Gupta, and today we are going to discuss on another topic, that is G6Pd Deficiency Anemia. So let's start. So G6Pd Deficiency Anemia. G6Pd Deficiency Anemia is the most common enzymatic deficiency of RBC, and the most common enzyme involved in this deficiency is Glucose-6-Phosphate Dehydrogenase enzyme, that is G6Pd enzyme. And the mode of inheritance is X-linked recessive disorder, and because it is X-linked recessive disorder, male will be more commonly affected than female. This G6Pd deficiency can cause hemolytic anemia, which can be both intravascular as well as extravascular hemolysis. Now coming to the etiology, there are various factors responsible for G6Pd deficiency, but the most important ones are drugs, infections and food. Among drugs, primaquine is the most common cause of G6Pd deficiency. It can also be due to various infections like bacterial infection, fungal infections, and fava beans may result in favism. And all these etiology, they are responsible for the formation of free radicals. These free radicals will cause increased oxidative stress on RBC. Now coming to the pathophysiology, there are two pathways that are responsible for G6Pd deficiency anemia. The number one pathway is Hmp pathway, that is hexose phosphate pathway, and it is also known as pentose phosphate pathway. So the goal of this Hmp pathway is to produce NADPH. This NADPH is required for glutathione reductase, fatty acid synthesis and cholesterol synthesis. This Hmp pathway occurs in cytoplasm of liver, mammary gland and adrenal cortex. And the another pathway is glutathione pathway. In this pathway there is free radical detoxification, which is done by reduced glutathione, and in order to detoxify the free radical, reduced glutathione requires NADPH, and this NADPH comes from Hmp pathway. And the main goal of this glutathione pathway is to form reduced glutathione, which will detoxify the free radicals. Now we'll come to the reactions. This glucose-6-phosphate comes from glycolysis, and this glucose-6-phosphate is converted to 6-phosphogluconolactone. So glucose-6-phosphate is converted into 6-phosphogluconolactone in the presence of G6Pd enzyme. So this is the rate limiting enzyme. This pathway converts NADP plus to NADPH. So NADP will be converted into NADPH during the conversion of glucose-6-phosphate that comes from glycolysis into 6-phosphogluconolactone in the presence of glucose-6-phosphate dehydrogenase enzyme. This NADPH is a reducing agent. Now we'll go to glutathione pathway. So this pathway is a glutathione pathway where reduced glutathione has to be produced in order to detoxify the free radical. The oxidized form of glutathione is converted into reduced form of glutathione. So in this process, oxidized glutathione is converted into reduced glutathione in the presence of glutathione reductase, where glutathione reductase will convert this NADPH back to NADP plus. Now the reduced form of glutathione will neutralize the free radicals. They will cause free radical and peroxide detoxification. So peroxide gets converted into water, so the RBC will survive. Now what happens in case of G6Pd deficiency? So when there is decrease in G6Pd enzyme, so when there is decrease in glucose-6-phosphate dehydrogenase enzyme, there will be decrease in NADPH production. So when there is decrease in NADPH production, So this will cause decrease in formation of reduced form of glutathione. So now this will cause increase in oxidative stress. This will cause increase in oxidative stress, so hemoglobin denaturation will occur, which will also cause misfolding of protein, and there will be RBC hemolysis. So there will be hemolysis of RBC, which can be both intravascular hemolysis or extravascular hemolysis. In G6Pd deficiency anemia, you will be able to see bite cells and hinge bodies. So bite cells, they are the most characteristic findings of G6Pd deficiency anemia. So these bite cells, it is also known as degmacytes. It is also known as degmacyte, and this portion is due to denatured protein. So this will look like as if a bite has been taken from the RBC, and this portion is actually a denatured protein. You can also see hinge bodies. So in order to stain the hinge body is best stained by supravital stain, that is crystal violet stain. In order to visualize these hinge bodies, you have to stain it by supravital stain, that is crystal violet stain. Hinge bodies can never be stained by Romanowski stain. Hinge bodies can never be stained by Romanowski stain. Now, what are the clinical features of G6Pd deficiency anemia? So there will be episodic intermittent hemolysis. So there will be episodic jaundice is seen. So episodic jaundice is seen. You will also see dark colored urine. And G6Pd deficiency anemia, features of chronic hemolysis will be absent. So there will be no splenomegaly and no cholelithiasis. So splenomegaly is absent and cholelithiasis is also absent. Now coming to the lab findings are suggestive of hemolytic anemia. So there will be decrease in RBC, increase in reticulocyte count, increase in serum LDH level, increase in indirect bilirubin, and there will be decrease in haptoglobin. Coombs test is negative. Coombs test is negative, and Coombs test is done in order to rule out immune mediated anemia. In peripheral blood smear, you will see bite cells and hinge bodies, and enzyme assay detects the level of G6Pd enzyme. So enzyme assay will detect the level of G6Pd enzyme, and this is the most accurate and definitive test. This is the most accurate and definitive test of G6Pd deficiency anemia, and it is done after 2 to 3 months of hemolysis, because young cells have normal G6Pd level, but as the cell becomes matured, G6Pd level goes down. So how will you treat this case? So for the treatment, you have to avoid the offending agents. You have to continue giving folic acid, and if there is massive hemolysis, then blood transfusion may be required. So this is all for today, and I will see you in the next videos. Thank you.